I'm going to talk about a wider way to look at the brain. I don't know what we have been looking at microscopes and ways to look at the cells. And I'm going to talk about MRI. I don't know if many of you know what's an MRI machine. So it looks like something like this. It has a magnetic field inside, which is normally between 1.5 and 7 Tesla, which is pretty high. So what happens when we put someone inside this machine is that the brain would react as a magnet. That means that we have these spins that comes from the hydrogen in our brain that are freely moving inside. And when the magnet starts reacting, all these spins would come to an organized way. So by measuring this kind of moving of the spins, we would know which is the tissue that we have in the brain. So let's imagine that I have one of these machines, and my question would be here, how does my brain look like? So can I use this machine to take pictures of my brain? And actually, the answer is yes. And you see here some brain pictures taken with one of these machines. In this example, these images are taken with an amperage sequence, so we can see the difference between brain tissues, mainly. So we can measure the density of gray matter, white matter, or we can even differentiate between different subcortical structures. For example, here, in this article, they used a very high magnetic field, and they could really draw the different hippocampal subfields. Or here, also with using these 7 Tesla machines, it was possible to differentiate these tiny structures within the brain. Also, with this kind of images, what we can do, and this is a very powerful tool, is to map the cortical surface, or all the cortex, into a surface. So we create maps like this one here, where each vertex is calculated using some kind of triangulation methods. These kind of representations are very useful in neuroscience because at first point, they allow us to look at these areas that are somehow hidden. So it's like inside the sushi, you don't know what's happening. But if you inflate them, you can see what's happening inside there. Another thing that it's more from the computational point of view is that we can just flat this surface, and we get to a 2D image, which is much easier to process for our computers. Finally, another very interesting thing that we can do with this is to calculate the cortical thickness, which is the distance. I see that some of the arrows have been displaced here, but it's supposed to be the distance between the white matter and the pile surface. That means that we can know how thick is our cortex, and we can know it at each point of this curve. So we pretend to have even higher resolution than the one we had with the voxels. So if we map this cortical thickness across one subject, we would get to something. Oh. There was an image here that I don't know. It's gone. We had the cortical thickness of one subject. That was just an example of how one of these maps would look like. But the thing is that if we put some subjects together and we analyze this cortical thickness across the entire surface, we can see, for example, which are the areas that have decreases with age. We know that our brain gets more atrophic with age. But in this first paper in 2004, the authors showed that there was a decrease in the cortical thickness in subjects that were around 80 years when you compare them with subjects of 20 years old. Later on, like 10 years later, we have seen that if we map the cortical thickness from very young subjects, like children, and we continue mapping it until the last years of life, we see how these changes are somehow continuous across life. So now we know how is the gray matter, and we know how is the cortex. But we might ask ourselves, can we look even deeper in the brain? So can we see how is the microstructure of the white matter of the brain? And yes, again, the answer is yes. We have another kind of MRI, which is the diffusion-weighted MRI. And using diffusion-weighted MRI, we can acquire a set of images which would look at the diffusion of the water in different directions inside the brain. So if we take all these images together and we approximate at each voxel how does the diffusion look like, we have some areas with anisotropic diffusion, meaning that it's more likely to have a fiber inside this area. So, and other areas where the diffusion is isotropic, meaning that the water can move freely inside this area. So with this kind of ellipsoids, we can map the areas where it's more likely to have a fiber passing through them. Going a bit further, if we put all these ellipses together, we can estimate the trajectories of the fiber. So we can get to maps like this one. That looks amazing, and it's like trying to follow all the fibers that we could see inside the brain. This diffusion-weighted MRI, or DTI, has been used, for example, to analyze how is the development of the brain during the first years of life. For example, here, in this article, it was shown that many of the tracks were increasing at the first decades of life, meaning that the fibers were getting more directed. So it would be an equivalent to say that our axons are getting to their way. And in healthy aging, we have the opposite behavior, where in some areas, which is not as clear as in development, but there are many areas where the axons become, or the white matter becomes less diffuse, so it becomes less directed. What else? We know how is the brain, we know how is gray matter and how is white matter, but we want to put all this machine working. So can we look at how does the brain work? And we can do this by measuring brain activity. So here we come to the third MRI modality I'm going to explain now. In this modality, we put the subject also inside the machine, and we acquire during a certain period of time. And we ask the subject to do something. This something can be something very easy, like finger tapping or some motor task. But it can also be something very complicated, like solving mathematical problems or do any kind of cognitive task or memory task. So during this time, we can measure how this is very huge compared to what we have been hearing before, but we can measure which areas of the brain are more likely to be active, to have more blood flow on them, because these areas would have an increase in this kind of signal that we are measuring with MRI. So the response to a stimulus would look like something like this. We get a stimulus to the brain, and then these areas get an activation for a while. Well, I will try to explain you this with an example. I don't know if it's... imagine that we'll put one subject into the scanning, and we ask him to perform a very difficult task for a while during these blocks. For example, we turn on the task at these periods. In other periods, we ask him to perform an easy task, something that would be very similar, but will be easier. Then, if we compare the activity, we can see how some of the brain areas are lightened during the performance of the difficult task, meaning that these areas are involved in this task. Going a bit further, we can also map areas that are specific for some stimuli. For example, here we see which are the areas that are active just for faces. Well, these are some examples. I'm going to pass this a bit quick. This is another tendency of FMRI, and it's becoming quite used in neuroscience. It's FMRI at rest. It seems a bit stupid at the beginning because we are trying to measure activity in a brain that's not doing anything, that's doing nothing. So we ask our subjects just to lie down for a while. And what we see during this while that they are lying down is that the brain is connected. So there are some fluctuations in low frequency that are correlated across all the brain. So then we can map things like this. So these areas will be connected to these other areas in the frontal, but they are very far away. So it means that something would be happening that makes them to be connected. Furthermore, if we just do this repeatedly, we can get to map a set of networks that are always in our brain. So meaning that our brain is always highly connected, even if we are resting or supposed to be resting, because you can never ask someone to rest without thinking in anything. But let's say spontaneous thinkings or spontaneous thoughts. So we have the brain, we have its activity, and now what can we do with this? So can we really answer questions related to neuroscience using these kind of tools? Because they look very good, they look very nice, the images are very cool, but what we want to know is how can these tools help to understand the brain? And I'm just going to show you some examples of people that have done these kind of things. So these maps have been obtained with EGI, and they are just a map of the connections. It can look like the map of a tube, for example, of the metro, because it maps the connections within the brain. And you can see that some of these connections are really more important than the other ones. That are the managers of our thoughts. And it happens that really these connections are correlated with the cognition or with executive functions of these people that were analyzed here. And in another example, in this study shown here, they used a sample of, I think, 400 twins or something like this. And they got to say that genetics and socioeconomic status were modeling the relationship between our structure and our intelligence. So it's kind of a 3 or 4 factor interactions. Other examples would come from FMRI. In this study was part of my thesis, my PhD thesis, we got to analyze a group of subjects that underwent a resting state, FMRI, and then they were asked to perform a task. So we got to say that the status of the brain during the time before performing the task was predictive of that task. So it means that the time you rest before performing something, your brain is actually doing some connections that are going to be used afterwards for the task. This is a more general study, the second one, and this was published this year, maybe 2 or 3 weeks ago. The authors here, this comes from Oxford, they have been mapping the whole brain, or the whole map of brain connections. And they associate some of these connections to what they call the positive traits. And they say that the positive trait is something that would make you good. So it could be something like having a good memory or having good skills, but it can also be having a good life expectations or even something like being happy. And they also got to the negative part of this. So having behavioral problems or having addictions or having anything that we can think about a negative trait would be bad, would be mapped in some of the brain connections. This is just a very nice picture, but just to put in common that for now we can make a map of our brain, as the one I showed you before, we can map how the structures work, and we can even go a bit further and map about how these functional networks are connected between them. So we get to a structural and a functional network. It's supposed, some people could think that this network and this network can be similar, and because it's the same brain, so they are related. But people are starting to think that structure and function are not that a perfect match. And we can't really think about it, because we only have one structure, and we have millions of functions. So the function needs to be a bit adapted to this structure. But these are some thoughts that come from studying the healthy brain. And now I get to my last question. What happens when something goes wrong in this system that's working? So how can we use MRI to understand diseases? Oh, there was another slide that was... Anyway, it's fine. I had a slide showing how is a brain, an histological brain of an Alzheimer's disease, and a reconstructed brain of an Alzheimer's disease. And you could see that really, we can see the atrophy of the brain. These are the different ways we have to map Alzheimer's disease, for example. So we know how is the cortex of an Alzheimer's patient, which are the areas that are most affected. We know how is the white matter of an Alzheimer's patient. And we also know how does it work. It doesn't work the same way as a normal subject. And it is very interesting to know that some of these changes are appearing even before the symptoms. So if we take a subject that is at risk for Alzheimer's disease, maybe his brain starts to work a bit different than a normal subject. So this is the point where neuroscience wants to go. So can we discover something that appears before the subject starts to complain or starts to have problems? And finally, these are some examples of other diseases. It's not only about Alzheimer's disease. So each disease would have its own pattern of changes. And we can map them very easily with MRI. And also in schizophrenia, it has been, it's not everything about neurodegenerative diseases. It's also about psychiatric diseases. And we can see, for example, here that the schizophrenia is a mix of genetic and environmental things. And it's good to try to get some conclusions of what's happening by one side and by the other side. And also in schizophrenia, doing this kind of maps, we see that the brain of a schizophrenic patient is much more chaotic than the brain of a normal subject. Meaning that, as you could know, a schizophrenic patient, so they have much problems in trying to organize his brain. And that's all. This is the group we are working with. And all these people are doing these kind of cool things with me. And time for questions if you have anything. Okay.
So we're running a little bit over time, but some quick reactions from the panel.
I was just wondering, since our brain is aging, do you think that would set a limit to our perhaps maximal lifespan?
I guess too, I think, but as all the human body, so I think I am starting to believe in what we call successful aging as well. So because we are also getting tools to analyze what happens in the brain of people that's getting old and that's still working well. So I think it's more that side than there is something that we cannot solve, that is that we will get old at some point. But I think we can just try to know what can make us being successful.
Your mapping of the brain is giving a view of how it works and how the diseases develop. Is this tool being taken into use when it comes to practice? If you need a psychiatrist or psychologist, do they use this data?
There are some data they can use, actually, because in functional MRI, you could, for example, if you have a patient that's not responding to some kind of stimuli, you can observe if his brain is responding to it, like in vegetative states and all of this. But all my work, or all what we are doing, it's more related to understanding how is the disease. So in the clinical day-to-day, I think it's very difficult to take one brain and decide. But it's always our aim, so